Thank you all for coming to part of the 60th anniversary of the founding of the biochemistry program by two individuals, George Villafranca and Ken Hellman, respectively in biological science and chemistry, who had the foresight in 1957 to found a biochemistry major at Smith, which, as I said at the lunchtime panel discussion, is probably one of the oldest biochemistry majors in the country. And we should be really proud of that. And also proud of all our alumni, some of whom we have with us today from 4 different decades of the program, which is great. Actually, one individual I worked with George Villafranca back in the 60s. And then we have others who worked with Professor Carr or me.
Exactly, you.
So this is the keynote address. I just want to remind you that tomorrow Erin is giving a talk on cryofluorescence and cryo-electron microscopy of viruses and how they enter cells. This is a burgeoning field, and if you recall, this year's Nobel Prize in chemistry went for the development of cryo-electron microscopy. This afternoon we have a talk by Erin O'Shea, but before I get into that, I have a number of people that I want to thank for putting this on. First, of course, is Kelly DeForest, our administrative assistant, who it couldn't have been done without her. Her logistics and everything was wonderful. First, I want to thank Professor Jameson, who also couldn't have done it without her, and our liaisons, who have done a remarkable job as well. And then I've got to mention that President McCartney wanted to be here, but since her regrets she couldn't come, but wishes us well in our 60th year and knows that we're going to have wonderful, and so far it has been a wonderful celebration. And the co-sponsors for this event are the Smith College Chapter of Phi Beta Kappa, Smith College Chapter of Sigma Psi, the Scientific Research Honorary Organization, the Office of the President of Smith College, and the Office of Alumni Relations. So, now with that out of the way, I have Erin O'Shea's advising question.
Oh, no.
And this chock-a-block full of all sorts of interesting
I need to sit down for this.
I don't think much time, but suffice it to say that Erin has created her own path to where she is now. She started off at Smith. She wasn't sure if she was going to be econ or biochemistry. Chose the better of the two paths, I may say, but took a year off after her sophomore year to go work at the National Magnet Laboratory on NMR of insulin, and it was in Cambridge. She just took the year off and went there and learned NMR. Really learned NMR because she learned it at the time when techniques as cozy, nosy, etc. were being developed. All right. So she came back, took my, the first time I taught biochemistry, the first time it was a 2-semester sequence. Erin was one of the guinea pigs in that class, but she managed to get through that. And she took a molecular biology course I taught that was really physical biochemistry. Historical reasons for that. And decided to do an honors thesis, but decided to do an honors thesis on work at the Magnet Lab and asked me if I'd be her Smith reader for it. And I said, sure, I'd be happy to. She started sending drafts, at which point I said, Erin, you've got to come teach me what cozy and nosy are. I had an idea of how NMR works, but I didn't know these techniques. So she came, and it was the first time I saw what a wonderful teacher she was, too, because she managed to teach those techniques to me. Well, she graduated in 1988, and then went off to MIT in the chemistry department to get her PhD in 2.5 years. While she was doing that, she had an offer from UCSF, University of California, San Francisco, for a faculty position. She decided that she wasn't ready for it and wanted to work in Tietjens' lab at UC Berkeley. So she asked them to put it on hold for a couple of years while she went and did a postdoc, which they said yes to, wisely. She went, did a postdoc, then went to UCSF and started going up the ladder of success in all sorts of manners and things and whatnot. Oh my God, it was incredible. I just want to say that for her PhD work, 2.5 years, 7 papers in 3 years, all in Science, Nature, JBC, things like that. 7 of them, okay? And to the point that she got a letter from Sir Francis Crick congratulating her on her paradigm-shifting structural analysis of transcription factors. As a graduate student. Okay. She then, I'm not going to go through all the stuff she did, but she's done incredible stuff. At UCSF, she became vice chair of the biochemistry and biophysics department, studying the role of randomness in cellular molecular interactions. Okay, then went on, was spirited away by Harvard to become the director of the Faculty of Arts and Sciences Center for Systems Biology, named a chaired professor. And in 2001 was awarded the National Academy of Sciences Award in Molecular Biology. And then in 2004 she was elected to the Academy, National Academy, as one of the youngest members ever. Now, remember, she graduated in 1988. This is 2004. Okay? Then she was named a Howard Hughes investigator, and in 2013 she was named vice president of Howard Hughes, vice president and chief scientific officer of Howard Hughes Medical Institute, and then was named president of HHMI, being the first woman to ever be named to that prestigious position, and also not unaided. All right, so she broke the glass ceiling in a number of ways, even there. All right, so I think I've said enough, other than it's fabulous to have her back again. Erin will share.
Okay. Thank you for that very generous introduction, Mr. Scordelis. And it's really too generous. I said some of this to the class earlier this morning, but I think it merits repeating. I really got a tremendous amount out of being at Smith for 4 years, both personally and it had an enormous influence on my career in many ways. And I mentioned this morning. I left here feeling really empowered that I could do anything I set my mind to, and that came from my experiences here. I also left with a great foundation in the sciences, and I said this morning, but one of the classes I took here with Mr. Scordelis, it may have been molecular biology. It was molecular biology. Where we were learning things, I remember at the time, they were interesting, but I wasn't sure I would work in this area or use the information. And of course, we all know Mr. Scordelis loves muscle proteins. And so we learned a lot about muscle proteins and the structures that some of the proteins in muscle have. And in particular, the one that I learned about that had an enormous influence on my career was this coiled coil structure that Francis Crick had proposed from some fiber diffraction data years before. But at any rate, I learned about that structure and a lot of the details of the structure. And that information enabled me to see something in a paper someone else published on transcription factors. And that insight that came from the knowledge I learned here at Smith in this class enabled me to publish the 7 papers he talked about and get a PhD in 2.5 years. And it's really not an exaggeration. So I guess the message is I'm deeply grateful for the education I got here, for the experiences I got here, for the mentorship that Mr. Skordillis and others have given me here. And I think it's a real lesson that the things you learn at the time, you may not realize their importance, but later on, you, in many cases, come to use the information in ways that you would never anticipate. So, so with that, I want to tell you now about 2 pieces of work from my lab that are really a continuation of what I talked about in the cell biology class, but I'll do this in a way so that those who, I know many of you were not in the cell biology class, who were not there will understand what I'm talking about. And I chose these 2 things to talk about because it's recent work from my lab. One of them is not published, and both of the pieces of work were done entirely by one graduate student, Anya Puzinska, and this represents her thesis work. And I think this is a sort of fitting to talk about her work in this context. So as this title says, which is not the same title I gave, I'm going to talk to you about clocks and tuners and sort of 2 strategies that the bacteria we work on use to thrive in an environment that's unpredictable. Okay, so as I said, I'll tell you 2 stories, and I'll start with this first one. And this story is focused on the function of the circadian clock in this bacterium called Synechococcus elongatus, which I'll tell you about in a minute. So first I have to explain, for those of you who were not around this morning, what a circadian clock is. And so these are endogenous molecular oscillators. They're mechanisms that cells in many, many different types of organisms have for keeping track of time. And they have 24-hour periodicity. And they're useful for an organism because they enable the organism to coordinate the organism's physiological state with the time of day. They enable the organism to schedule physiological processes to occur at certain times of day. The circadian clocks, as I said, are ubiquitous. They're found in organisms ranging from humans to rodents. The fruit fly here, for which the Nobel Prize winning work on circadian clocks was awarded this year to Jeffrey Hall, Michael Rosbash, and colleagues for decoding the genes that are involved in circadian control in this model organism. Clocks are also found in plants, in some fungi, and in certain bacteria, like the one depicted here. And this is a cyanobacterium. So what are cyanobacteria? Well, they are unicellular, rod-shaped bacteria. They're obligate phototrophs, so they rely on photosynthesis to generate ATP. They're thought to be the ancestors of chloroplasts in modern-day plants. Many of the cyanobacterial species have a circadian clock. They're responsible for a significant fraction of photosynthetic activity in the Earth's oceans. They're very easily grown in the lab, which is why we work on them, and they have excellent genetics. So they're genetically tractable, allowing us to unravel problems in biology at a molecular level. So this cyanobacterium, we study the species is called Synechococcus elongatus. It doesn't really matter, but it's the simplest model organism in circadian biology. It's a single cell, as I said, and the clock has all of the general features common to all circadian clocks. So this system has a core timekeeping mechanism that we call the core clock. This clock, as I said, keeps time with 24-hour periodicity and enables the organism to schedule its physiology to take place at certain times of day. And the major way in which the clock schedules physiology is by controlling expression of genes at the level of transcription. And I'll tell you a bit more about this in the next few slides. So we call this output from the circadian clock. Output is the control of the expression of genes. And it turns out that the majority of genes in the genome of Synechococcus elongatus are under circadian control. So this is really prevalent type of regulation in this organism. And finally, to be useful, the core clock has to be able to receive input from the environment to enable it to synchronize with the predictable changes in the environment, such as the light-dark cycle. And so here I've shown input as in the form of light, which is a dominant cue for this organism and for others, such as humans. And so mechanisms exist by which the organism can take input from the environment and alter the phase of the clock such that the phase of oscillations of the clock inside the cell are in sync with the phase of oscillations in the environment, right? And so these are features common to all circadian clocks and again found in this simple model organism. So I'll just remind you, well, and tell the people who weren't around this morning that in Synechococcus elongatus, the core clock turns out to be a post-translational oscillator. And this is perhaps the most remarkable feature of this system that I won't say much more about, but it turns out that these three proteins in a test tube, when mixed together with ATP, are sufficient to generate sustained oscillations, just like you see inside the organism. Oscillations in the phosphorylation state of this protein called Chi-C. Okay, so that experiment that was done by the Kondo lab is really the clearest demonstration that these three proteins are necessary and sufficient to generate circadian oscillations. And so this clock, unlike what is thought to be true for the fruit fly clock and the human clock and essentially all others, this clock is thought to be but based on a post-translational mechanism. Well, I should say this clock is based on a post-translational mechanism. I think it hasn't been proven clearly that no post-translational mechanisms exist in other organisms. Okay, so this is something remarkable about the cyanobacterial circadian clock, the fact that it's the oscillations derive from this post-translational mechanism that I talked about in depth this morning. And again, for those who weren't here, I just said this in words, but the phosphorylation state of one of the three clock proteins called Chi-C oscillates with circadian periodicity, and this is how time is encoded and read out in the circadian clock in this organism by decoding essentially the phosphorylation state of Chi-C. And the details of this don't matter for what I'm going to tell you about today. This is merely background. Okay, so now I'd like to turn to this issue of output, which will really form the basis of most of what I'm going to tell you in the first part of this talk. So I told you that a major way in which the core clock controls physiology in Synechococcus is by controlling expression of genes, most of the genes in the genome. And it turns out that there is a single transcription factor that acts as a master regulator of circadian gene expression. And what I mean by this is that activation of this factor, even in the absence of the clock, is sufficient to trigger a cascade of gene expression whose timing looks just like the timing observed in the natural circadian cycle. Okay? So again, what I mean by this is if you activate RpaA, this transcription factor, even without the clock present in the cell, the genes are turned on in the same order as they are in the normal circadian cycle. Okay, so this is amazing. And it turns out that RpaA doesn't do this alone. It only binds to 100 genes, but there are thousands of genes in the genome of Synechococcus controlled by the clock. And so I won't talk about this today, but RpaA, it turns out, controls a cascade of other transcription factors that lead to this differential timing of expression of genes in the genome. Okay, so RpaA, the most important part for you to take away is that it's a key, it's a master regulator of circadian gene expression. It's controlled through regulation of its phosphorylation state, indicated by this little red ball, which is meant to represent phosphorylation. It is active when it is phosphorylated, and it can bind to DNA, the little squiggly lines. And when it binds to DNA, it can turn on expression of genes. The phosphorylation state of RpaA is controlled by the clock in a way that we understand, and if you're interested, you can ask me later. It's not really relevant for what I'm going to tell you about today. So, as is true of Chi-C phosphorylation, the activity and phosphorylation state of RpaA oscillate with circadian periodicity. And specifically, RpaA phosphorylation peaks right around dusk, that is when the sun sets, okay? And then declines during the night. Okay, so RpaA is maximally active here at around dusk. All right, and so really, I set this up by saying I wanted to talk to you about the function of the circadian clock in this organism, Synechococcus elongatus, the cyanobacterium. And really, more precisely, the question we want to answer is, does the circadian system confer a benefit? And turns out the answer is yes, or I wouldn't be telling you this. And then, given that it confers a fitness advantage, how does it do this? I mean, why is having a clock, if you're a cyanobacterium, why is having a clock beneficial? Okay, and that's the first question I'm going to answer today in the first part of this talk. All right, so how do we know the circadian system confers a fitness advantage? And that comes from some work done some time ago in Carl Johnson's lab at Vanderbilt. And what he did is a really simple experiment. So he took cyanobacterial strains, here growing in these green tubes, that have different circadian clocks that have different periods, right? So here, the period on the left is, much less than the period on the right, right? And he wanted to know whether these strains with different periods have a different fitness relative to one another. And so the first thing he did is to grow these strains that have clocks with different periods in constant light conditions. Okay, and we can do this in the lab. I told you the organism's an obligate phototroph. It requires photosynthesis to generate ATP, and they're perfectly happy growing in constant light. And the circadian clock keeps time even in constant conditions. This is something true of all circadian clocks. So in constant light, there's no difference in fitness dependent on this difference in period of the clock. But now, if you instead grow the organism in alternating light-dark conditions, where the period of oscillation of the light-dark cycles matches the period of oscillation of one of the clocks, then the strain whose circadian clock has a period matching that of the light-dark cycle has a substantial fitness advantage over the strain containing a circadian clock that is not matched to the environmental variation. Okay, and so this was really the first indication that in this organism, the clock confers a fitness advantage. And now the issue is really, how why? Why does it matter? So, and that question can be really reduced to the one I've outlined on this slide, and that is, what processes does the clock control to give a fitness advantage in an oscillating environment, but not in a constant environment? All right, and so to address this question, what we did is to analyze mutants in the clock. And I'm going to tell you about the one that's the most informative, and that is this mutant RpaA, which I told you is the master circadian regulator. So we can make a strain that lacks RpaA. We delete the gene. The strain is perfectly viable in constant light conditions. That's what's shown on the left here. So we just measure the density of cells, right, on the y-axis as a function of time. And you see the two strains. The black one is the normal strain, which we call wild type, and the red one is the RpaA mutant. You see they grow with the same growth rate in constant light conditions. And now on the right, if we grow the cells in alternating light and dark, the dark is signified by these gray bars and the light by the yellow bars. So if we grow the cells in these light-dark conditions, as we call them, now you see there's a difference in fitness between the two strains. And it turns out that RpaA mutant strains have a viability defect in light-dark conditions. And we know this from a number of experiments. It was first shown some years ago by the Kondo lab. And you can see clearly in this data that the RpaA mutant has a fitness defect. And the fitness defect is restricted to these alternating light-dark conditions. So now there's a problem here with the interpretation of the experiment I just showed you. So what we'd like to conclude is RpaA must be performing some function in these oscillating conditions that reveals something about the function of the circadian clock, right? And about the genes controlled by RpaA. But there's a problem, and the problem is as follows. Something I haven't told you is that the circadian clock, via this transcription factor RpaA, controls circadian gene expression. But it turns out that some of the genes controlled by the clock are the clock components themselves. All right, so that's what's meant by this transcription feedback. So the clock controls, via RpaA, expression of the clock genes themselves. So now what happens when we delete RpaA, the transcription factor, now there's no feedback, and now we abrogate expression of the clock components. So now the clock doesn't run, all right? So, and this is true. In the RpaA mutant, the clock doesn't run. So now we have a problem, right? We don't know whether the viability defect I showed you is a consequence of aberrant transcription, that's kind of what we want to study. RpaA-dependent transcription. Tell us the genes that are important for viability in oscillating conditions. But maybe, instead, the viability defect we see in the RpaA mutant is just the result of stalling of the clock, right? And so to distinguish between these two, we did the following experiment. So on the left is just the wild type, that's the normal strain, where RpaA, in its phosphorylated form, is promoting transcription of two of the clock genes, Chi-B and Chi-C, which are in this operon. Okay, and now when RpaA isn't present, it can't promote transcription, so there's a defect in expression of Chi-B and Chi-C. So we can rescue the defect in expression, and we call this the clock rescue strain, by now replacing the promoter, the regulatory region of the Chi-BC operon, with a different regulatory region, one that is insensitive to RpaA and to the circadian clock. Okay, so this is a constitutive promoter that we identified that drives expression of the Chi-BC operon independent of the function of the circadian clock. Okay, and that's why we call this the clock rescue strain, and we characterize this strain, but I won't tell you more about it. And now we can use the clock rescue strain to ask whether the viability defect is a consequence of clock stalling or of deletion of RpaA per se. And it turns out to be the latter. So here, now we compare two strains, the clock rescue strain, in which the cybc operon is under the control of some promoter regulatory region that's clock independent, and the same strain, the clock rescue strain, now lacking RpaA. And now we see the same viability defect. The clock rescue strain grows fine in alternating light-dark conditions, but if we delete RpaA in the background of the clock rescue strain, now there's a viability defect again. So in both of these strains, the clock is running fine, right? Even though RpaA is gone, the clock runs fine because we restored expression of cybc, and we still see this viability defect in the RpaA mutant, and this indicates clearly that the viability defect is not a consequence of clock stalling. It must instead be due to some defect, presumably, in the RpaA mutant and likely in genes regulated by RpaA. Okay, and so if RpaA is really important for viability in light-dark conditions, then we might expect that active RpaA, even in the absence of the clock, might be sufficient to maintain viability in light-dark conditions. And so what we did is to make a strain in which we delete the clock and delete RpaA, and then introduce one of two forms of RpaA. Either this D53E form, E is glutamate, and this mimics the phosphorylated form, remember the red ball, or RpaA in which D-aspartate 53 has been replaced by alanine, which mimics the unphosphorylated state. And then we asked whether either of these strains can restore viability in a cell that otherwise lacks the clock and lacks wild-type normal RpaA. And what we found was a clear result, and that is that the phosphomimetic, this glutamate mutant of RpaA, restores viability, but the alanine mutant does not. And so from these experiments, we conclude that active RpaA is necessary for viability of the cyanobacterium in light-dark cycles, right? If we delete RpaA, even in a strain in which the clock is completely functional, then we have a defect in viability. And it's also sufficient to restore viability in light-dark conditions. And now the question is, what does RpaA control that's actually important for viability, right? Can we understand why the RpaA mutant has this viability defect and use it to infer what the important function of the clock is? What functions of the clock are required for viability? And so to address this question, we did a simple experiment. I'm not going to show you all the data, but we used high-throughput sequencing to analyze systematically defects in expression in the clock rescue strain compared to the clock rescue strain lacking RpaA. And so we're looking for genes that are defective in their regulation in the RpaA mutant strain. Okay? And we got a very striking result. I already said that. And that is, there were a set of genes, and they all turn out to be in the same kind of pathway that I'll show you on the next slide, that have a gross defect in expression. So now this is RNA. We're measuring RNA levels in the RpaA mutant compared to wild type. And remember, this is this clock rescue background to make the experiment clean. And so the clock rescue is like the wild type situation that's shown in gray, and the RpaA mutant is shown in brown. And what you can see is that in every case, there's a gross defect in the expression of these genes. And in the wild type strain, they're expressed at actually quite high levels. Okay, so what are these genes? Well, as I said, they all turn out to be involved in a common pathway, and that is central carbon metabolism that occurs in the dark. They're involved in utilizing glycogen via glycolysis and the oxidative pentose phosphate pathway to generate ATP during darkness when the cells can't generate ATP via photosynthesis. Okay, so I've mapped on here the enzymes shown in red and the points in this pathway in which they act. And so this suggests that one important function of RpaA is to control the expression of these genes, which in turn enable this organism to switch from deriving ATP from photosynthesis to deriving ATP from the degradation of glycogen in the evening. And consistent with this idea, if we delete these enzymes that I've shown you, these are indeed enzymes that are controlled, whose genes are controlled by RpaA, what you can see is the strains on the left are viable in constant light conditions. But on the right of each of these two strips, you see that the cells can't grow in alternating light-dark conditions, consistent with the idea that these enzymes are important for growth in the alternating light-dark conditions. Consistent with the picture I've shown you, not only is the RNA level defective in the RpaA mutant, but if we measure enzymatic activity for 3 of these enzymes, we see gross defects in enzymatic activity in the RpaA mutant, shown in these red bars, compared to the wild type strain shown in gray. All right, so the first major conclusion I want to leave you with is that in the RpaA strain, the activity of glycogen utilization pathways is compromised. And this led us to ask whether energy levels are lower in the RpaA mutant cells at night, since I told you that the function of these pathways involved in glycogen degradation is to enable the cell to generate ATP from stored carbon sources utilized during the evening when they can't do photosynthesis. And so what we did is to measure energy charge by quantifying the levels of ATP, ADP, and AMP, and then calculating this parameter called energy charge that just reflects the utilizable high energy ATP and ADP equivalents. And you can think of this as like a battery, right? When the energy charge is high, the battery is full, and the cell has a lot of energy to utilize during the night. And so what we see is very clear. So on the left is shown this parameter energy charge. You can think of like the battery as a function of circadian time, measured for both wild type in the black, that's the normal case, and the RpaA mutant shown in red. And what you see is in constant light conditions, there's no difference in energy charge. And in contrast, in these alternating light-dark conditions, what you can see is that although the wild type can maintain energy charge over time, the RpaA mutant over successive light-dark cycles dramatically loses energy charge. And so the RpaA mutant strain has a defect in the maintenance of energy charge in the dark, and I don't think this is surprising at all given these defects in glycogen utilization that I showed you previously. So the next thing we asked is whether the RpaA mutant strain has normal levels of glycogen. And so first I need to remind you or tell you that during the light, glycogen is synthesized. Carbon skeletons derived from primarily from the Calvin cycle are then fed into a pathway that's used to generate glycogen, a storage carbohydrate for this organism that can then be utilized at night. And so we measured glycogen accumulation in wild type and in the RpaA mutant. And what you can see is a very dramatic defect in both oscillating light-dark conditions, now shown on the left, and in constant light, shown on the right, in the RpaA mutant. There's a dramatic defect in glycogen accumulation that takes place during the day. Normally, the wild-type cells during the day here in yellow rapidly accumulate significant stores of glycogen, which are then utilized during the night and then reaccumulate again during the day. And I'll just tell you that we know this defect in glycogen accumulation is not the result of RpaA directly controlling expression of genes involved in glycogen formation. We measured that. So we don't understand the molecular mechanistic nature of this defect in glycogen accumulation. It's clearly RpaA dependent, but is not a direct effect of the activity of RpaA as a transcription factor on genes encoding enzymes involved in this process. But suffice it to say that we have another defect in this RpaA strain. It doesn't accumulate glycogen during the day. And so now we have two defects. RpaA mutant cells can't make glycogen. I just showed you that. And first I showed you that the RpaA mutant cells can't burn glycogen. They can't utilize it. I mean, there could be many other defects in the RpaA strain. But if these are the two important defects, we should be able to engineer a strain to correct these defects. And if these are the important defects, these two, if we correct the defects, we should restore the viability defect. All right? And it turns out we do, or I obviously wouldn't be telling you this. And I'll just tell you in the next couple slides how we corrected these defects. So RpaA, done obviously in the RpaA mutant background. So RpaA mutant cells can't make glycogen. And so what we did instead to enable cells to form glycogen stores and break it down is to engineer sugar importers. So we express this transporter GalP from another organism that enables the cell to uptake glucose and then form glycogen and utilize this glucose directly. RpaA cells can't burn glycogen, and I told you the nature of this defect is that RpaA is regulating the expression of these enzymes that are involved in glycogen utilization. And so we can get around this by putting each of the genes that's defective in the RpaA mutant under the control of regulatory regions, promoters, that are RpaA independent. Okay, and so this is the strain here. It can get a carbon source inside the cell that it can utilize because it expresses this GalP transporter that allows it to uptake glucose. And it expresses all the enzymes that are defective in the RpaA background, and this is an RpaA mutant cell, under the control of a promoter that's not RpaA dependent and that's not clock dependent. Okay, and so now in the absence of glucose, so there's no glucose, so now the cells don't have both defects corrected, right? The strains don't grow. None of the combinations of strains. So RpaA mutant alone, with the transporter only, with the enzymes only, or with both. None of them grow. But now if we add glucose, you see only one strain really grows well, and that's the strain that has the enzymes, expression of the glycogen utilizing enzymes restored, and GalP so that the strain can uptake glucose. And so and furthermore, in this strain shown on the top here, energy charge is restored in that strain. And so we conclude that the two major defects we identified in the RpaA mutant are two major functions controlled by the circadian clock required for the viability of this organism in oscillating conditions. And those are the ability to generate carbon to be utilized during the nighttime, and enzymes that are involved in the degradation of those carbon stores to enable the cell to produce ATP under conditions of darkness where it can't generate ATP via photosynthesis. And finally, I'd like to return to this result from the Johnson lab here about the inability or about the difference in fitness between these two strains that have different periods. And if you recall, I told you the strain whose circadian period matches the period of oscillations of the environment has a fitness advantage over the one that does not match that of the environment. And now we can understand why. What's happening in the strain whose period matches that of the light-dark cycle is that it can appropriately switch metabolism from utilizing photosynthesis to generate ATP to utilizing stored carbon sources to generate ATP, whereas the strain with a mismatch performs this metabolic switch at the inappropriate time. Okay, so in the last few minutes, I want to tell you about the second part, which I hope is shorter. And this concerns a signaling molecule that enables cells to tune global gene expression in response to environmental perturbations. And so this signaling molecule is an old one called ppGpp. It's a derivative of guanosine diphosphate, GDP. It has two additional phosphates that are derived from ATP, adenosine triphosphate. ppGpp has been well characterized in a number of bacteria and also in plants, and it's involved in signaling stress, which is generally referred to as the stringent response in bacteria. And so it turns out that there are two enzymes, SPO-T and RelA, that are involved in the production of ppGpp. They're ppGpp synthases, and they utilize guanine nucleotides and ATP to generate ppGpp. One of the enzymes, SPO-T, also has hydrolase activity, so it can break down ppGpp to generate GDP and pyrophosphate. So in the field, ppGpp is thought to do many things, but one thing that it's thought to regulate is directly bind to and regulate the activity of RNA polymerase to influence the types of genes that are expressed, and specifically to switch gene expression towards a stress response and away from housekeeping functions. Okay, so I want to tell you about how ppGpp signaling is used in cyanobacteria. There's only one enzyme in cyanobacteria that's involved in generating ppGpp, and that is a SPO-T homolog, depicted here, which has both synthase, it can make ppGpp, and hydrolase, it can break it down, activities. So the first thing we did is to delete SPO-T, and it has only a minor growth defect in constant light conditions. But as shown first by David Savage's lab, it has a major defect in growth in alternating light-dark conditions. And now you can see the connection to the previous part of my talk. So SPO-T, like RpaA, has a defect in growth really only in alternating light-dark conditions. So it's required for viability in these conditions, and we'd like to know why. Why is ppGpp required for viability in the dark or in alternating conditions? And so one thing, again, that we saw and that the Savage lab also saw is that ppGpp levels, shown in black, increase dramatically when cells are switched from light, that's this little yellow strip here, to darkness, the gray. You see they increase rapidly and then decrease over time in darkness. In constant light conditions, what we noticed is that ppGpp levels are above the detection limit. So in the bottom here is the SPO-T mutant. And you see that in the wild type, the normal strain, there are levels of ppGpp detectable above background. So what we see is that in light conditions, ppGpp is present at this low level but detectable. And in darkness, ppGpp accumulates very rapidly when cells are exposed to darkness. And so what we want to know is what's the function of ppGpp in the light and in the dark? And is the increase in ppGpp in the dark required for viability? All right, so what's the function of the basal level of ppGpp? That's the level that's present during just constant light conditions in cyanobacteria. And so what we did is to look at gene expression in the SPO-T mutant in light conditions. And so what I'm showing you here is gene expression on the y-axis in the SPO-T mutant, that's not working, and in the wild-type strain on the x-axis. Each dot here represents a gene, and the dashed line is where all the points would fall if the expression in the wild-type and the SPO-T mutant were exactly equivalent. And what you can see is all the dots, most of the dots, are shifted above the dashed line, meaning that the expression of almost all the genes is higher in the SPO-T mutant than in wild type. Okay, so that's peculiar. So lack of ppGpp leads to a global transcriptional increase. And we can measure this by measuring transcription rate in bulk with a radiolabel with radioactivity. The translation rate is also increased in the SPO-T mutant, and so is cell size. Okay, so one function of this low level of ppGpp that exists in light is to reduce transcription, translation, and reduce cell size. And now we want to know what's the function of ppGpp in the dark. And so again, we delete SPO-T and then look at what happens. So first, I want to show you what happens to gene expression when cells are shifted from light, which is the first column here, to darkness, which are all the other columns. And so this is some heat map where each row is a gene. Each column is a different time point. I told you all the way on the left is before shift to darkness. And then all these times are times following the shift to darkness. Blue means the genes are turned off. And so what you can see, I told everybody this morning, is there's this massive transcriptional shutoff that occurs when cells are put in darkness. And this happens, this is known to happen in cyanobacteria. And so we asked if this still happens in the SPO-T mutant, and you can see there's a clear defect. The shutdown eventually happens, see here at 12 hours where everything is dark blue, but you see all along the way here, it happens a lot more slowly than in the wild type strain. So ppGpp is required for this dark-induced transcriptional shutdown. That's what I just said. And so now we've looked at this situation where the cells lack SPO-T, so they don't have any amount of ppGpp. And what we really wanted to know is what happens in this basal level strain, right? So is the basal level of ppGpp sufficient to restore any of the phenotypes in darkness? And there are only two phenotypes I told you about, the dark-induced transcriptional shutdown and the viability defect. So we want to know, is this low level of ppGpp enough to correct those defects in darkness? And so we have to engineer a strain that produces low levels of ppGpp, because in the wild type, remember, darkness makes ppGpp levels go very high. And so the way in which we did this is to express RelA, an enzyme from E. coli that makes ppGpp, but that is not sensitive to the dark, under the control of a constitutive promoter. It's not clock dependent, and we did this in a background lacking SPO-T. So the only way the cells can make ppGpp is by this enzyme RelA. And now we confirm that it behaves as expected. So that's shown on the right here, where we measure ppGpp levels as a function of time. So the wild type strain that has normal SPO-T and doesn't have any of this rigmarole on the left is shown in black. When it's grown in constant light conditions, it has low levels of ppGpp, the black line, and as soon as we shut the lights off, ppGpp levels go very high. All right, now the engineered strain, which is lacking SPO-T, SPO-T minus, but having RelA, this E. coli enzyme. That's shown in the green color, and what you can see in the green color is it has low levels of ppGpp in light and in dark, comparable to the low levels in the normal wild type strain present in light. Further, if we mutate the active site of RelA to make it catalytically inactive, it has no expression of ppGpp. And we can zoom in on these low levels and show that in the RelA strain in green and the normal wild type that has SPO-T, they have similar low levels of ppGpp. Okay, now we can do the experiment. We use this SPO-T minus, rel A plus strain and put it in darkness and ask whether that corrects any of the defects that we observed in the SPO-T mutant. And so first I should show you that with respect to gene expression, now here's wild type and here's SPO-T minus, rel A plus, and you see this rel A plus has gene expression that's basically indistinguishable from the wild type. It also corrects the cell size defect that we saw in the SPO-T mutant if we put in rel A. All right, so now we ask, can the basal levels rescue the defects in the dark? And the first thing we look at is this transcriptional shutdown, wild type shown on the left. And you see there's still a defect in the SPO-T minus rel A plus. So these basal levels aren't sufficient to cause transcriptional shutdown in the dark. And what this implies is that the elevation in ppGpp levels, this signaling molecule, are required to shut down transcription in this organism in the dark. But now we got a surprising result when we looked at viability. And now here in the SPO-T minus rel A mutant, what you can see is that the viability is restored, and it's dependent on the catalytic activity of the introduced rel A. So this is surprising. The massive increase in ppGpp levels that occurs in the dark is not required for viability in this organism. And so what we conclude is that ppGpp is present at these low levels in light conditions, in normal growth conditions, in the absence of stress. And this molecule plays an important role in tuning cell size and global translation and transcription rates in the organism. The elevation of ppGpp is required for transcriptional shutdown, but not required for viability. The only thing required for viability is the low level of ppGpp. So ppGpp is used to tune cell physiology. It tunes it via this mechanism I told you about in light and also in darkness. And so I'll just finish, so that's good, by reminding you the one person did all the work, one graduate student, terrific graduate student in my lab, Anya Puzinska. And so I'll stop there, and I'd be happy to take any questions. Oh, yeah, okay. That's a really great question. What controls SPO-T? So we don't know, but I'll tell you what things we do know. So we looked at a lot of different stresses and other chemical perturbations. And the only perturbations that result in an elevation, SPO-T dependent elevation in ppGpp levels are molecules that disrupt the photosynthesis itself. So it is likely that some product downstream of photosynthesis, electron transport, is involved in signaling to SPO-T. But we don't know the molecular mechanistic basis of how that works, and we don't know what molecule it is. The other thing we know is that SPO-T localization in the cell is regulated. So it is inactive. When it's inactive, not producing ppGpp, it's sequestered at the poles. And when it's active, it's diffuse in the cytoplasm. Yeah. Yeah, but that's a super interesting issue. We don't know the answer. Yeah. Yeah. Yeah, so I didn't explain this at all. So RpaA is activated directly by the clock. And so the clock is functioning independent of light or dark. And what happens is that certain phosphorylated forms of Chi-C bind to and regulate the activity of two histidine kinases that phosphorylate RpaA. They regulate its phosphorylation state, One can also remove the phosphate, but there are two histidine kinases that we identified, and they interact directly with the clock proteins. They're controlled directly by the clock proteins, and they act directly on RpaA to regulate its phosphorylation state. Yes, that's something we know quite well, and that mechanism operates independently of, as I said, light or darkness. It's timed only by the circadian cycle itself. So you are right in that activation, meaning phosphorylation of RpaA, takes place only during daylight. Yeah. Yeah. Oh, yeah. Okay. Yeah. Go ahead. I keep looking back there because somebody way back there.
Oh, on the scatter plot of
Yeah.
genes regulated by SPO-T wild type versus SPO-T mutants,
Yeah.
are you interested in any of the outliers that don't seem to follow that same pattern of regulation?
Yeah. I'm sure we looked at those genes. It's a really good point. I can't tell you what they are now and whether they're meaningful. It's a good question. I just simply don't know the answer off the top of my head. It's a good question. Yep.
I was wondering if you could put the graph up of the RpaA mutant again in the cell viability assay.
RpaA.
Oh,
yeah.
RpaA
Okay.
mutant, that's a long way back. Just any one of those. I don't think it matters which format. I just noticed that the cell viability started to drop off around 72 hours. And I'm thinking you're thinking that's like it's reaching some sort of critical point.
Yeah.
Like ATP
Yeah, no,
depletion. But I'm thinking more again that more than that. I mean, I can understand that that's reaching some critical ATP depletion there, after that time frame. But I don't know what the turnover time is.
No, this is a really good point. I didn't make this clear at all. So here we're measuring optical density, which is not a good proxy for viability. Okay, so this isn't the right experiment to clearly draw the conclusion I drew above. So what we really do, and I just didn't go through all this minutia, is to plate samples as a function of time, either after placing in constant darkness or in this alternating regime. And we can see the loss of viability even after short incubation in darkness. You're totally right that this isn't, I kind of did a little sleight of hand. I would have buried you in like some boring data. Anyway, yeah, it's a good point.
But I'd also expect
Yep.
viability to drop off more during the dark, and it doesn't appear to really do that. It doesn't seem to be light dark dependent beyond 72 hours. I'm just thinking if it's the lack of glycogen and the lack of utilization of glycogen in the dark.
Yeah, I agree. I would have expected eventually it to sort of like totally peter out. I don't know off the top of my head why that doesn't happen, but what I do know is that if you put the cells in constant darkness, they very quickly become absolutely inviable, as defined by if you remove an aliquot and plate them on a plate, you don't get any colony forming units. Yeah. Yep.
So why is there a transcriptional shutdown in the darkness
Yeah.
of the S. elongatus if it doesn't
Yeah.
decrease viability?
Yeah, no, that's a great question. Why is there transcriptional shutdown in darkness? And I suspect that that is a way the cell has to preserve, energy stores and energy in general. It's not transcribe all these genes. I mean, there are it's big gene expression program. But the truth is we don't really understand why. And we certainly don't understand exactly how that works. I can tell you that, so I told you that this significant increase in ppGpp is required. And the other thing we know is that many of the genes whose transcription is rapidly shut down or most dramatically shut down, the polymerase is paused. It's sitting at the promoter of the gene. Okay, it's not transcribing the gene, but the polymerase is just sitting there in a ppGpp dependent way. And when you turn on the lights, those polymerases are mobilized very quickly. So I suspect some of the transcriptional, and the genes that are in the category I just described with the paused polymerase are involved generally in translation. There are a lot of ribosomal subunits and other proteins involved in translation. So a translation consumes an enormous amount of energy in the cell. And so I suspect, I mean, I'm just speculating here, that the transcriptional shutdown, which involves to a large extent these genes involved in translation, the polymerase is paused there. The cell is trying to conserve energy, but it's doing so in a way such that when the lights get turned back on, and the name of the game if you're a bacterium is to then grow fast, the polymerase is sitting there ready to transcribe the gene, and it does transcribe the gene. Yeah, so that's all we know. We don't know more about that. It's a very good question. Yeah?
Can I ask a follow-up to that has to do with the energy level of the cell? Is there anything to be gleaned either from plotting the ATP concentration and ADP concentrations themselves, or looking at the whole nucleotide profile, which would then impact the deoxyribonucleic acid pools in that process?
That's a great question. That's another one like the genes off the diagonal. I should know, but I don't know. I can't even make something up. I just don't know. Yeah?
So, how general is this nighttime metabolism?
A lot of organisms use glycogen as energy stores, but I suspect that this, this dramatic switch that takes place is special to organisms like this, and there are other organisms like this that undergo this dramatic reprogramming of metabolism from photosynthesis-based generation of ATP to utilization of carbon stores. So there are many bacteria that have that switch, but beyond bacteria, like for example, plants, I don't know the answer to that. I'm sure it's known. Plants definitely utilize stored carbon. Yeah.
On a related note,
Yep.
did you ever ask the bacteria to come up with another strategy to survive by a genetic screen?
We didn't, and I bet you would find interesting things that way. That's a good experiment. We've not tried that. Yes, and then look at what you get, what mutations you select for. Yeah, it's a good question. I don't know. I don't know the answer, but it would be definitely worth doing. Okay, one more. Okay, before Mr. Scordilis gets down here.
I don't know anything about bacteria. Just wondering why you use the same time interval for the light and dark treatment.
So here, that's a good question. I didn't really explain this. So here we're just trying to mimic what happens in a normal day. It's about 12 hours of light and about 12 hours of dark. Of course, it varies with the season and whatnot, but we just mimic what's happening in a normal day with 12 hours of light and 12 hours of dark. Of course, it's not a very good mimic, right? Because it's not like when the sun comes up, it's not like turning the lights on, and that's what we're doing here. And when the sun goes down, it's not like turning the lights off. And in a normal day, you might have clouds, the intensity of the sunlight might vary. And that's not at all accounted for here. We've done a recent study where we looked at more natural light intensities, and we chose light intensity profiles by using light intensity measurements from a detector on the top of a building at the University of Wisconsin, and then we subjected the cyanobacteria to those profiles by using a set of programmable LED lights. And that turns out to be quite interesting. But your point is well taken that this is meant to mimic the natural day, but it doesn't do a good job of mimicking the natural day. But experimentally, it's easy, so that's why we do this. Yeah.
Thank you again very much.